Hi, everyone. Welcome back. So, we are going to be starting up our genetics notes. So, hopefully, you looked in your notes folder and you found our newest set of notes entitled Genetics. So, genetics is really fun. I know it seems like this has a lot more notes than some of our past units, but genetics is cool because we can see, how traits are inherited and how they're expressed. So, just like always, when you open up your notebook, you have your title. Slide number 2 has your vocab, and some of this you may know because you did go over some basic genetics when you were in middle school. And then the learning objectives. So learning objectives will have some stuff that we haven't gone over yet. And so, we're going to spend a little bit of time on this unit, but it's going to be really fun. This is one of my favorite units. So let's go ahead and get started. So all of today, basically, or all of this video rather, is going to be a review. So most of this stuff you probably remember, and so I'm actually going to go through this pretty quickly. And if you need extra help, let me know, and then we can arrange for some tutoring time. So first off, I have this portrait right here of this esteemed gentleman. His name is Gregor Mendel. So he was known as the father of genetics. So what Gregor Mendel did is he studied pea plants and determined that traits are passed from parent to offspring through something called genes. Okay, so he was actually pretty lucky. So when he looked at the pea plants, he actually was able to look at 7 different traits, and all 7 of those traits only have, 2 expressions, so to speak. So, for example, for flower color, you either had purple flowers or white flowers. For, let's say, like the pea color, you could have yellow peas or green peas. So these traits that he looked at were very simple, okay, because there were only 2 choices to have, either of those traits. So something else that he did, he called variances, there we go, oops, of those traits alleles, okay? So alleles is going to be one of your vocab words, and I'll explain here what an allele is, okay? So you've probably heard that we have dominant and recessive traits. So dominant and recessive are the allele types for that gene. Okay, so if I look right here on a chromosome, I can see I have 2 chromosomes here. I have a blue chromosome and a red chromosome. So the blue chromosome has an allele for purple flowers. Okay, so purple flowers is dominant, and so that is the dominant allele. White flowers is recessive. So this red chromosome has an allele for white flowers. So when I'm talking about these alleles, my dominant is the allele that is always expressed if at least one is present. Okay, and so we also represent this with a capital letter. Okay, and I don't remember how I got this to fit the first time. Let's see here. Oh, that's not going to work. Okay, so let me do this. Okay. There we go. I think that's actually what I did before. Okay, so represented by a capital letter. I'm just going to put that represented a capital letter. Okay, all right. So my recessive allele. Okay, so the recessive allele is normally hidden by the dominant allele. Okay, so normally hidden by dominant allele. Okay, so the only way you can see the recessive allele is if you have 2 of the same. Okay, so if not, the dominant allele is going to overpower it. And so this one is represented by a lowercase letter. Okay. So we know that we have 2 chromosomes. You get one from mom, one from dad. Okay, so that's what makes up an organism. So because of that, we have, an allele from each chromosome. So for our alleles, they can be homozygous, so two of the same. Okay, so for example, two dominant or two recessive. So, like let's say that for these purple flowers, we're going to use the letter F. Okay, so maybe we have big F, big F. It's going to be homozygous dominant, or little f, little f. Okay, we can use whatever letters that we want. So this little f means that we have two recessive alleles, and that's the only way you're going to see that recessive trait. If we have a heterozygous set of genes, then that means we have one of each. We have one dominant and one recessive. Okay, so one of each allele. Okay, so if we use our letter F again, okay, that means we have big F, little f. So remember, if we have a dominant allele present, that means the recessive allele is going to be hidden. We're not going to see that. So if we're still going with flower color, that means that this individual would still have purple flowers because they have a purple allele. Okay? So next, let's go ahead and set up how we can figure out what our offspring would have. So what I have right here, okay, so I have a Punnett Square, and I can see I have a true breeding green pod and a true breeding yellow pod. So right here, okay, so what true breeding means is that the individuals are homozygous for their trait. Okay, so sometimes we call these purebred. Okay, so if you ever hear of something being a purebred, that means it's true breeding. It's homozygous for the trait. Okay, so if we do this right here, okay, so if we do cross two true breeding, all of our offspring are going to be heterozygous. Okay, so when two true breeds are crossed, all offspring will be heterozygous. Okay. So when we're looking at our Punnett Square, we could see that we have a green true breeding pod. Okay, so we know it's homozygous, so it's big G, and a yellow true breeding pod, that's little g, little g. These two individuals are known as our P generation. Okay, so that P stands for parent. Okay, so my text does not fit. Make this a little smaller. Means I need to make this one smaller too. Okay, there we go. Okay, so this is my parent generation. Okay, this is my original generation. All of these individuals in here, these are my offspring of that parent generation. So we call that the F1 generation. Okay, so the F1 generation are going to be the offspring of the P generation. Okay, so, and I can see this F1 generation, they're all heterozygous. Now, if I take this F1 generation and I let them interbreed, okay, so I know each individual is heterozygous. So if I come over here to this Punnett Square, I have two green pea pods because they have at least one dominant allele. And so when I separate this out, I have big G up here, little g down here, big G up here, little g down here. So if I let them cross, okay, and I look at my offspring's genes, this is going to be my F2 generation. Okay, so it's my second generation. All right. So I can see now my yellow, my yellow characteristic comes back. Okay, so I end up with 75% that are green, okay, and then 25% that's yellow. So the colors are my phenotypes. So my phenotype means a physical trait. Okay, so yellow, tall, oops, green, okay, so things like that. Okay, those are physical traits. Those are physical descriptions. Okay. My genotypes are the letters that represent those physical descriptions. Okay, so the genes represented by letters. Okay, so for example, big G, little g, little g, little g. Okay, so I can see right here I have 3 different genotypes. Okay, so I have one green pea plant that's big G, big G. I have 2 that are big G, little g. And then I have 1 yellow that's little g, little g. Those are my genotypes. But when I'm talking about a phenotype, I have 3 out of the 4 that are green and then 1 out of the 4 that's yellow. So in order to figure out what our offspring are going to be, I'm going to use a Punnett square. So this is a table that helps, predict, you're not going to be able to see because of my camera. There we go. Okay, so helps predict the offspring's traits and genotypes based off the parents. Okay, and I think I put my apostrophe in the wrong spot. There we go. Okay, so if we know what the parents are, we're able to figure out what our offspring are. Okay, so let's go ahead and practice really quick. Okay, so, and I have a little GIF here that, walks you through step by step how to do a Punnett square, just in case you've forgotten. Okay. So what I have right here, I have a blank Punnett square. Okay, now here's my problem. In guinea pigs, the allele for short hair is dominant to the allele for long hair. I'm going to cross 2 heterozygous guinea pigs, okay, and the letters I'm going to use are L, big L, little l. So I know that these guinea pigs are heterozygous and they're both long-haired, okay, because they have 1 big L present. So it doesn't matter where I put which parent. Okay, so this one right here, okay, I'm going to say is parent 1. Okay. This one right here is going to be parent number 2. So both parents have the same genotype, and I'm going to split it up because each parent only gives half of the genetic information. Okay, so this letter right here is, oops, and I got rid of it. Okay, that letter right there is big L, and this one's going to be little l. Okay, should actually switch that. Let me do that right there. Big L, okay, little l. Okay, same thing for up here. Okay, big L, little l, because my parents are the same. So next, okay, the letters that are on top are going to be filtered down. The letters to the side are going to be filtered to the right. So if I match up my 2 letters, I have big L, big L. I'm going to match up these 2 letters, big L, little l. I'm going to match up these 2 letters, big L, little l. And then lastly, I have little l, little l. So if I'm looking at my genotypes and phenotypes, I can create a ratio. Okay, so I have a genotypic ratio of 1 big L to 2 big L, little l to 1 little l. I can also do that in fractions. I could say that it is 1/4 big L, 1/2 big L, little l, and 1/4 little l. I could also do it as a percent, 25% big L, 50% big L, little l, and 25% little l, little l. Okay, so you just have to look at the problem and see what it's asking for. But my phenotypic ratio, okay, so remember, long hair is dominant to short hair. So that means that 3 of my guinea pigs, 3 out of the 4, are going to have long hair. Okay, so I have 3 long hair to 1 short hair. Okay. So again, I could do percentages. I can do fractions. Okay. All right. So let's do some practice really quick. Okay. So I'm going to go ahead and pause this, and go ahead and try to do this so that way you can check your answers. Okay. All right. So number 1, will the puppy have a spotted coat? Well, if I look right here, okay, so I have a chromosome from the mom dog and a chromosome from the dad dog. So if I look for spotted coat, I can see spotted coat's represented by big A, and it's a dominant gene, so that means I only need 1. So I can see the mom gave an allele for a spotted coat. So that means yes, the puppy has a spotted coat. Does the female dog have a spotted coat? Well, you only need one dominant allele to have that, so the answer is yes. So now this is where students get confused. Does the male dog have a spotted coat? Well, if we look at the male dog's chromosome, we have a little a, but he only gave half of his genetic information to the puppy. So for all we know, his other chromosome could be a big A. So we don't know. We can't tell from this information. Okay, so I'm going to put IDK, okay, because we don't know. Okay, so what's the texture of the puppy's coat? So if I look at hair texture, I can see wiry is big T, silky is little t. Okay, so I see I have a little t from the female dog and a big T from the male dog. So we have at least one big T here. Okay, so we know that the puppy's coat is wiry. Okay. So why can't you completely describe the puppy's parents even though you can describe the puppy? Okay, so we only have, half of the parent's genetic info. Okay, we have the puppy's full genetic information, but for the parents, we only have half. All right, our next one we're going to practice Punnett squares. Okay, so we have right here, we have some phenotypes, purple flowers and white flowers. Okay, purple flowers are dominant to white, so we know if we have big P, that's purple. Okay, big P, little p is also purple. And little p is white. In guinea pigs, short hair is dominant to long hair. Okay, so big H is long hair. Big H, little h is also long hair. And that means, oh wait, I did this wrong. Okay, so short hair is dominant, not long hair. Okay, so that means that big H, and big H, little h are short hair. Okay, good job if you got that. In guinea pigs, black eyes are dominant to red eyes. Choose the letter combination for each phenotype. So we can pick whatever letter we want. I'll use the letter B since black eyes are dominant. So our two genotypes that are possible are big B, or big B, little b. The only way you can have red eyes is if you have little b. All right, last two things. We're going to do two Punnett squares. Okay, so they're already set up for you. So all we need to do is fill them in. Oops. Okay. So little b. Okay, remember, we bring the ones down from the top, and the ones over on the left, we bring to the right. Okay. So if I look, how many guinea pigs will have black eyes? Black eyes are dominant, so I can see I have 2 out of the 4 that are black eyes and 2 out of the 4 that are red eyes. So I can put 2/4, or I can condense that and put 1/2. And it's going to be that way for both. Same thing here. Okay, so my big B's are going to come down. My little b's are going to go to the right. So that means that in this case, all my guinea pigs are going to be big B, little b. Okay, so something to note. I do make sure to put my dominant allele first. Okay, it's not necessarily a rule, but it's something that is helpful when you're trying to figure out the genotype. Okay. So how many guinea pigs will have black eyes? So 4 out of 4, I could say 100%. I could say, 4 out of 4, which is what I'm going to do. Okay, but I don't have any that have red eyes, 0 out of 4. Okay, so no red-eyed guinea pigs from that one. Okay. So hopefully this helps. Let me know if you have any questions.